Hello, my name is Josh Mahlios, and I'm a Senior Marketing Product Manager for Milteni Biotech. And thank you for joining this webinar that focuses on the latest technologies and resources available for immuno-oncology researchers. Today, we will be focusing on highlighted products for immune subsets that are increasingly recognized as some of the key regulators of the tumor microenvironment. These include the good, NK cells, the bad, T regulatory cells, and the ugly, macrophages. The agenda for today is as follows. First, I will briefly provide some background information about Milteni Biotech as a pioneer in immuno-oncology research and therapies. Next, we will discuss the importance of reproducible tumor processing and sample preparation. We will then talk about the fundamentals and advantages of MAX technology for isolating rare cell types and our automated cell separation solutions. This will be followed by highlighting the breadth of our cell culture and cell analysis portfolios. And finally, we will discuss our translational and clinical offerings. Empowering discovery and advancing therapy. These are the words that most accurately define our company's mission, which is to improve scientific understanding and medical progress by providing solutions that advance biomedical research in cellular therapies. And as a company, we are able to achieve this mission by our ongoing commitment of bringing newer and smarter technologies to immuno-oncology researchers who are paving the way for longer and healthier lives. Miltoni Biotech was founded in 1989 by Stefan Milteni, and the basis of MAX cell separation is the column technology developed as a part of his diploma thesis, with this technology being patented in 1990. The potential for MAX technology to become a revolutionizer in the fields of gene and cellular therapies became obvious, and the Clinimax CD34 reagent system for selecting hematopoietic stem cells from donor apheresis was developed. The clinical evolution of the company has continued for over 20 years, including setting up a GMP manufacturing site in Teterow, Germany. And in 2014, the FDA gave approval for CD34 isolation using the Clinimax together with the Clinimax CD34 reagent. What started out as a diploma thesis has since grown into a global company of more than 3,000 employees and well over 20,000 scientific publications citing MAX technology. Annually, more than 2,500 patients receive cell products manufactured using Milteni Biotech technologies, allowing us to recognize our company mission of advancing biomedical research and cellular therapies. While initially leveraging MAX technology to advance stem cell therapies, much of our attention is now focused on the field of immuno-oncology. So, why are we as a company so focused on this scientific field? In part, it is due to the fact that nearly 10 million people die from cancer every year, meaning that cancer is the cause of 1 in every 6 deaths, and it is one of the largest health problems facing the world. In light of all the recent medical progress, how is cancer mortality still increasing over time? There are a number of contributing factors, and primarily these include an ever-growing and aging population. So we can fully expect cancer to remain one of the leading causes of death for the foreseeable future. So what are some of the most common types of cancers that we encounter? As shown in the figure below, a majority of cancer diagnoses and deaths in both men and women can largely be attributed to solid tumors, this being dominated by lung, prostate, breast, and pancreatic cancers. It is for this reason that the scientific community and Milteni Biotech have been keenly focused on this aspect of oncology, which is the targeting of solid tumors. The formation of solid tumors involves the coevolution of neoplastic cells, along with the extracellular matrix, tumor vasculature, and various immune cells. The conditions within the tumor play a critical role as to whether the immune system itself or clinical treatments are successful in eliminating these types of cancers. The dynamic tumor microenvironment varies drastically, even throughout the same lesion. And these various cell types within the tumors can actively influence therapeutic responses and shape resistance. So what are some of these key players within the tumor microenvironment that we will be focusing on? Now, as I just mentioned, The composition and characteristics of the tumor microenvironment vary widely and are important in determining the anti-tumor immune response. For example, certain cells of the immune system, including natural killer cells, dendritic cells, and effector T cells, are capable of driving potent anti-tumor responses. However, tumor cells often induce an immunosuppressive microenvironment, which favors the development of immunosuppressive populations of immune cells, and these include myeloid-derived suppressor cells and T regulatory cells. Understanding the complexity of immunomodulation by tumors is important for the development of immunotherapies, and we as a company are poised to provide all the tools needed to better understand these complexities. Okay, so let's start talking about actual research and the associated challenges that are faced by those working on the bench. We will begin with the importance of tumor processing and sample preparation and the solutions that Milteni Biotech has to offer. Tumors are comprised of a mixture of immune and cancer cells, extracellular matrices, basal membranes, and vasculature. When dissociating solid tumors, there are three primary goals. The first being to reproducibly dissociate your tumor into a single cell suspension. The second being the preservation of cell surface epitopes. And the third is to maintain cell viability and functionality. Speaking of solutions, what happens when you receive a tumor at the end of the day and you aren't prepared or appropriately scheduled for all the necessary downstream processing and analytical steps? Fortunately for researchers, we literally have a solution for this very situation. Our Max tissue storage solution has been developed to allow for the optimized storage of fresh tumor samples without showing background effects like activation or the induction of apoptosis. It has been tested and validated on a variety of human and murine tissues, including not only tumors, but skin, heart, spleen, brain, and skeletal muscle. The result is the preservation of viability, cellular composition, and gene expression, giving you the confidence that you can adequately prepare for all the necessary steps to properly process your sample when you are ready. When it comes to dissociating tumors, a combination of mechanical disruption, whether it be mincing, straining, pipetting, or the use of bead mills, along with enzymatic digestion. Now, these steps are often user and provider dependent and can result in rather variable results. Our tumor dissociation kit has been developed for the gentle and rapid generation of single-cell suspensions from human tumor tissue. Our kit is optimized for high yield of tumor cells and tumor-infiltrating lymphocytes, while preserving important cell surface epitopes. This kit is ideally suited for the time-saving and reproducible preparation of single-cell suspensions in combination with our GentleMax dissociators, which provides the mechanical disruption. When it comes to processing your sample, just doing a mechanical dissociation by itself can result in the loss of cell populations which are closely attached to the tumor interface. These tumor-reactive T cells are often lost and not recoverable. By using the tumor dissociation kit, we enable you to identify these rare cell populations. This is demonstrated in the first panel, where you can see that significant populations of interest are not visible using traditional methods of tumor dissociation, whereas these PD-1 positive, LAG3 positive, or TIM3 positive populations are readily observed while using our specially formulated tumor dissociation kits. To build upon this, we tested whether the three enzymes used for enzymatic digestion affect the integrity of cell surface epitopes, scoring from stable, moderate sensitivity to strong sensitivity. Our results show that a vast majority of the more than 200 epitopes tested are well preserved during these enzymatic treatments. If your marker of choice is sensitive, contact technical support for tips on how to adjust the enzyme combinations to preserve your epitopes of interest. As an additional example, I will briefly discuss the importance of epitope preservation in the context of CD25, the high-affinity IL-2 receptor that is one of the defining surface markers for T regulatory cells. As mentioned, CD25 is a key cell surface marker that is used to identify Tregs, though this epitope is particularly sensitive to enzymatic digestion, making the identification and subsequent isolation of these cells difficult, if not impossible. By adjusting the enzymatic combination of our tumor dissociation kit, you are able to preserve this critical marker, giving you a much more representative and useful readout of the cellular composition of the dissociated tumor. There are additional surface epitopes that are important in the identification and isolation of both NK cells and macrophages that need to be considered when dissociating your tumor samples. Whether it's CD107b for macrophages in murine samples or anti-NKP80 for NK cells in human samples, you can remain confident that you are actually obtaining the necessary information to proceed with your downstream analysis. For a full listing of all the surface markers that might be of interest to you, please visit our website to download the comprehensive cell surface epitope preservation list. Once you have successfully, and more importantly, reproducibly processed your tumor samples into single-cell suspensions, the isolation of target cells is often the next step that researchers take for their downstream applications. First, I will give a little background information about Max technology and why we have become the gold standard in cell separation, especially for rare populations. Max technology is based on three key components: Max microbeads, the reagent, Max columns, and the Max separator, the magnet. Together, they enable magnetic separation of cell populations based on surface antigens. This technology is a fast and gentle method for the isolation of viable and functional cells by labeling epitopes with specific antibodies conjugated to superparamagnetic beads. In order to separate your cell populations of interest, only three easy steps are needed: magnetic labeling, where an antibody conjugated to tiny superparamagnetic microbeads that bind to a receptor or protein on the cell surface; magnetic separation, where the cells are passed through a column that is positioned within the magnet. The cells labeled with the microbeads are retained within the column, while everything else is washed through in a gentle way. The flow through is called the negative fraction, containing the cells without the ligand for the antibody or antibodies that were being used. In the elution step, step number 3, the column is removed from the magnet, enabling the magnetically labeled cells, so the positive fraction, to be washed out. This can be done either manually with a plunger to gently push the cells out, or it can be automated using a vacuum to pull the cells out, or increasing the flow speed of the buffer in the system. All three methods are gentle for the cells and separate the cells into negative and positive fractions with exceptional purity and high viability. A frequently asked question is, why do we use columns in the first place? The magnetic separators in column-based and column-free systems have a comparable magnetic force, but the difference between the systems is the column. And this column is filled with small iron spheres. Each iron sphere passes the magnetic force on to the next over a very small distance, thus amplifying the magnetic force by 10,000-fold and ensuring that the magnetic field is constant throughout the column. This system allows for the minimal labeling of cells with microbeads to retain the target cells in the column without relying on epitope saturation or large beads that can interfere with downstream applications such as cell culture or flow cytometric analysis. The iron spheres have a cell-friendly coating that allows for the cells to run freely through the column and can be washed thoroughly to remove cell debris and contaminants. All of this contributes to the fact that Max columns allow for highly effective cell separation with minimal effects on cells. If you would like to learn more about the technology itself, its benefits, and to see some data, feel free to have a look at the webinar recording, Select the Best with Max Technology, on our website. The link will also be included in the recording of this webinar, or just search cell separation in the video library on our website. By leveraging the advantages of Max technology, we have developed a number of kits for the isolation of NK cells, Tregs, and macrophages from both human and mice. And while I won't go into the details of these various kits, you can rest assured that we have the products and solutions for the isolation of these key cell types for most any researcher. When it comes to isolating cells, You can choose the cell separation option or options according to your needs, whether it's a manual process for a limited number of samples, all the way up to a fully automated high-throughput cell separation instrument. Milteni Biotech has you covered. Now, while manual cell separation certainly has its place in certain day-to-day operations, our focus now is on automation. But why do we actually need automation? What is the advantage? Automation reduces hands-on time and frees you up to work on other tasks in the meantime, tasks that cannot be automated. It minimizes variation between different users in the lab, or even good and bad days of an individual scientist. And of course, it minimizes costly human errors. With standard protocols and minimal user variation, you can be sure that the effects that you're seeing are due to your assay rather than artifacts. In short, automation reduces redundant experiments and allows you to focus your time where it matters. There are two instruments that I would like to highlight at this point. If you're interested in isolating multiple samples in parallel, the Multimax family is a perfect fit for this. 24 magnets allow you to process between 1 and 24 samples in one run, or split your large sample between multiple columns. This is achieved by using the LS, LD, or whole blood columns, or the multi-24 column block for the easy handling of 24 samples, or one very large sample, like an entire leukopak. The Multimax 24 is a semi-automated instrument and a good fit for many labs that value easy handling but don't mind manual pipetting. However, if you want full automation and maximum throughput, you'll love the Multimax X, which integrates a Multimax into a liquid handling system. As mentioned, the Multimax X is the instrument for the high-demanding, high-throughput cell processing lab. Samples are labeled and isolated automatically, both for PBMC generation or cell subpopulations from tumor samples. All programs are tailored to match your needs, so we don't leave the lab until you're satisfied with the protocol and the results. For many researchers, once you have isolated your cells of interest, the real work begins. This often starts with the culturing of target populations for downstream functional assays. So in the next section, we will talk about our cell culture portfolio, which includes activation, expansion, and functional assay products. Our Max cell culture and stimulation portfolio comprises a specialized and versatile range of cell culture media and reagents for cell stimulation, expansion, and/or differentiation. Products are available for human and mouse primary cells, including immune, stem, neural, and tumor cells. Many of these products are available in both research and GMP quality, allowing for the smooth translation into the clinic. First, we will discuss the media options that we have from our cell culture portfolio. For scientists that are working with T cells, our TexMax medium has been designed for high-performance T cell growth, high cell viability, and consistency under serum-free conditions. It has been optimized for the serum-free cultivation and expansion of human and mouse T cells and Tregs. Our TexMax medium has a defined formulation enabling reproducible applications in human and mouse cell cultures. It is produced without animal-based derived components, but contains pre-selected human serum albumin, stable glutamine, and phenol red. Our research-grade medium has been developed based on our TexMax GMP medium, again allowing for the smooth translation into the clinic. For researchers working on NK cells, we offer our NK-Max medium, which is the perfect solution for NK cell cultures and expansion from PBMCs or isolated NK cells. The chemically defined xeno-free composition ensures reproducible results. Expanded NK cells are fully functional and can be used in any downstream application, such as killing assays. If you're starting your expansion from PBMCs, the unwanted growth and expansion of T cells and NKT cells is minimal. For researchers that are looking to establish primary tumor cell lines, our cancer cell culture medium is optimized for the efficient initiation and expansion of human tumor cell cultures from primary and xenotransplanted tumors. With standardized serum-free formulations, our Tumor media offers optimal conditions for the generation of primary cell lines by enhancing cell proliferation without inducing any genomic changes. The cells will maintain their heterogeneity and tumorigenic potential, improving in vitro models for cancer research and drug screening. Tregs are often functionally analyzed in vitro by a so-called suppression assay. For this purpose, Tregs are co-cultured with T responder cells at different ratios in the presence of a polyclonal stimulus. Tregs alone show a hypoproliferative response, whereas T responder cells alone can show a proliferative response. By co-culturing Tregs with T responder cells, you can often see a reduction in the incorporation of either thymidine or the dilution of the cell tracking reagent in the T responding cells. This suppression assay is performed with a dilution ranging from a ratio of 1:1 to 8:1 of responder cells to Treg cells, as outlined in the figure below. This product allows you to evaluate the functionality of your Tregs, not just based on phenotypic analysis of surface markers or cytokine secretion. In this next section, we will be discussing some of the critical aspects that need to be taken into consideration when phenotyping your cells of interest, especially in the context of the tumor microenvironment. Syngeneic mouse tumor models represent a gold standard to analyze the effects of immunotherapy, as they possess a fully competent immune repertoire. Such analyses routinely rely on the characterization of tumor-infiltrating leukocytes and their phenotype. However, the amount and composition of TILs is highly variable and often very low, complicating the flow cytometric analysis of individual subpopulations. To make things more difficult, traditional hybridoma-derived antibody clones used for flow cytometric evaluation of TILs show a tendency of nonspecific binding to immune cells, such as myeloid cells, via naturally expressing the Fc gamma receptors, or induced expression of Fc gamma receptors on T cells in the context of the tumor microenvironment. This can lead to an inaccurate and biased estimation of TIL populations. To avoid Fc gamma receptor mediated background binding, blocking reagents, including commercially available immunoglobulins, are used. This adds additional complexity to the analysis, as there's no defined consensus on the best titer and type of blocking reagents that is supposed to be used for complex tissues like tumors. In addition, blocking reagents increase staining time and hinder the ability to scale up or automate multi-sample analyses. Here I'm describing the benefits of using our Reaffinity recombinant antibody clones for the analysis of different TIL populations. Reaffinity antibodies provide several benefits over hybridoma-derived antibody clones. For example, a specifically mutated human IgG1 Fc region that abolishes their ability to bind to the Fc gamma receptor. This enables more reliable and reproducible flow cytometric analysis. So I'd like to take a brief moment to describe a little bit about our Reaffinity antibodies. Our Reaffinity antibodies are recombinant antibodies that provide superior lot-to-lot consistency and purity compared to mouse or rat hybridoma-derived monoclonal antibodies. They have been recombinantly engineered to produce highly specific antibodies that require no Fc blocking. And additionally, they have the same IgG1 isotype, requiring less isotype controls. The manufacturing process of Reaffinity recombinant antibodies ensures high performance based on the following aspects. They are derived from a defined DNA sequence, which encodes only one type of heavy and light chain, ensuring high antibody purity. Production is under biologically and chemically defined standards that result in high lot-to-lot consistency. Analytical, biochemical, and cell-based testing of every lot ensures you're going to get the highest quality, most reproducible antibody possible. All Reaffinity recombinant antibody clones contain the same specifically mutated human IgG1 sequence as their constant region. This offers the possibility to work with only one type of isotype, reducing the complexity of experiment planning and saving time as well as money. Hybridoma-derived monoclonal antibodies, on the other hand, are composed of antibody isotypes derived from different species and therefore require multiple isotype controls. This complicates not only the management of reagent inventory, but also panel design when setting up experiments. All Reaffinity recombinant antibodies contain a specifically mutated human IgG1 Fc region, which abolishes their need to block or to bind Fc gamma receptors. This allows for background-free analysis and eliminates the need for additional blocking steps, such as using an FCR blocking reagent. One of the product highlights that I'd like to draw attention to in our cell analysis portfolio is our MDSC Detection Kit. Now, myeloid-derived suppressor cells are a heterogeneous group of mononuclear myeloid cells with different immunosuppressive as well as disease-promoting functions. In healthy volunteers, they're found in relatively small numbers, but can expand considerably in pathological conditions. Depending on the type and stage of disease, the MDSC population might be present in different maturation phases. Validated, unambiguous phenotypic markers for MDSCs are still lacking, and therefore various combinations of specificities are used for their identification. The MDSC Detection Kit is intended to be used for the standardization and simplification of flow cytometric analysis of subpopulations of MDSCs described thus far. For greater flexibility, the product configuration also allows the extension of the analysis panel with additional specificities of interest, examples being CD10 or CD66B. The MDSC Detection Kit has been designed for the detection of circulating MDSCs and cell suspensions of PBMCs, and might be used for the analysis of other starting materials such as whole blood, lysed whole blood, or single cell suspensions from human tissues such as tumor cells. So finally, I would like to briefly mention a few key aspects of our translational and clinical offerings for those that are looking to take their research to the clinic. As an innovative biotechnology company, we at Miltoni Biotech offer cutting-edge technologies and high-quality products to support your immuno-oncology research. Whether you are interested in studying immune cells or cancer cells from the tumor microenvironment, or cellular models for the validation of novel immunotherapeutic agents, check out our applications page for the respective cell types, whether they're engineered T cells for research, macrophages or MODCs, NK cells, or tumor-infiltrating leukocytes, to find relevant application data and protocols based on our immuno-oncology products. Moreover, we offer clinical-scale immuno-oncology products for the easy transition of your research workflows into the clinic. I'd like to thank everyone for attending this webinar, and for additional information, please visit our booth here at the LabRoots Immuno-Oncology virtual event, or our website for all the tools and solutions needed to advance your potentially life-saving research. Thank you, and keep up the good fight.